Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Okay, thank you for the time. And before starting for the presentations, let me introduce myself. So my name is Dwi Setiorini. I'm a junior researcher in the Research Center for Biology, Indonesian Institute of Sciences. And the titles of my presentation is Analysis of the Metal Accumulation and Phytochelatin Synthase gene expressions in Saccharum spontaneum as regarded function for remediation of the lead contaminations. And I would like to start with the introductions about the heavy metal pollutions. So we know that the heavy metal pollutions currently is being a very serious problem in the world, and these metal contaminants generally come from the various anthropogenic activities in many sectors, such as in industry, in domestic, in agriculture, and also in the medical sectors, by which then these activities will produce a huge amount of the disposal waste that can spread widely in the environment. And even though some of the heavy metal they can serve as an essential element for the living things, but most of them they are very toxic, especially if they're present in the high concentrations. And this toxicity is due to the formations of the complex compound in the cell. And one important thing that we have to know regarding to the presence of the heavy metal contamination in environment is once they enter in the environment, they cannot be decomposed. They will persist and accumulate in environment, and meanwhile they can also affect and pollute the food chain, which is in turn it will directly induce the increasing of the various health problems in the human. And among the heavy metal contaminants, lead is defined as the one of the most common hazardous contaminant. And naturally, lead presents as a microelement in a trace amount in the soil, in the water, and also in organism, even though the biological functions of the lead itself in the living organism remain unknown until now. Lead persists in the environment in the forms of the stable ions, and of course it's quite difficult to remove the lead from the soils when it has strongly bounded in the soil matrix. And now we talk about the phytoremediation, which is also well known as green remediations. So the phytoremediation is determined as an in situ remediation technique by using the hyperaccumulator plants to remove the harmful contaminants from the environment. This technique is also a kind of the low-cost technology that can be applied in contaminated site. So the phytoremediation technique emphasizes in the abilities of the plants to absorb the contaminant from the environment optimally without disturbing their growth. And as a subject used in this study, Saccharum spontaneum, which is a kind of the grass plants and belong to the Poaceae family, can survive to grow in the polluted area where no other plants that can be cultivated there. And it means that Saccharum spontaneum has the potential to be used as an accumulator plant to clean up the metal pollutants in the environment. The in situ research experiment was designed to identify the abilities of Saccharum spontaneum plants as the lead accumulator. And the study was conducted to analyze. The first one is to analyze the lead accumulations, and the second one is to analyze the Phytochelatin Synthase gene expressions. And the observation was performed in the both of root and the shoot of Saccharum spontaneum plants under the various concentrations of the lead treatment. And the results show that after 8 weeks, there was an increase of the lead levels deposited in the soil, which was in line with the increase of the lead levels of the treatment. And in this study, the increase of the lead treatment corresponds with the increase of the lead levels in the plants. And however, the roots of the Saccharum spontaneum plant show the higher lead level as compared to the shoot in the same lead treatment. And the presence of the metal pollutants in the soil greatly influence the metal contents of the plant that's grown on it, unless there is a mechanism that inhibits the absorption of the metal by plants. And the presence of the metal toxicity in the soil is being the one of the stress factor for the plants. And this stress factor can affect the plant's growth and even productivity. And whoever the plant species which is in the study we use Saccharum spontaneum plants has the ability to absorb the toxic element, resulting in the increase of the metal concentrations in the biomass plant. And the results in the Phytochelatin Synthase genes indicated that the expression of these genes was higher in the wood than in the shoot, with an enhanced range of 1.3 to 3.5-fold reductions as compared to the control. And these results correspond with the results of analysis of the lead accumulations in the plant, where the higher soft lead accumulations was detected in the wood than that of in the shoot. And the mechanisms of the heavy metal tolerance in the plants is regulated by the complex networks of the genes, which one of these genes is a phytochelatin synthase. The phytochelatin synthase genes can produce the enzyme which can be found in a wide range of the organisms, such as in plants, in fungi, in nematodes, and also the groups of algae. This enzyme functions to catalyze the formation of the phytochelatins, by which the phytochelatin itself can serve as a chelators to chelate the heavy metals in the vacuole and decrease the cell damage. Therefore, it is very important for the heavy metal detoxification in the plants. And for the conclusion in the study, the first one, the lead treatment subjected to the closed medium was able to induce lead accumulation in the Saccharum spontaneum plants. And the second one, the lead accumulation in the roots was higher than in the shoot. And the third one, the phytochelatin synthase gene expression in the root showed 1.3 to 3.5-fold reductions, which were higher than the shoot under lead treatment. And the fourth one, the lead accumulation in the woods correlated with an increase of phytochelatin synthase gene expressions in Saccharum spontaneum plants. That's all, and thank you very much for your attention.